project. First conceived in the mid-1980s with the idea of locating and mapping all of the genes that make up the human genome. Nobody really knew how many genes there were. Some estimates had it as much as 100,000 different human genes. The task was immense. 6 billion bases make up the DNA of the human genome. We know that they're made up of 4 different nucleotides: adenine, thymine, guanine, cytosine, but no idea of the order. So by the time 1990 started, many laboratories around the world decided to share the workload and start to sequence the human genome, which was a very slow and laborious process to begin with. So during the course of the years, new technologies were invented and new techniques were devised. Computer power and new machines such as DNA sequences enabled the process to be automated and the speed of sequencing to increase. The cost of all this is now estimated to be about £3.9 billion. That's at 2018 values. So it was completed ahead of schedule, and completion took place in 2003 for the human genome. Other organisms had also been sequenced at this time, lots of parasitic worms, for example. What did we find out? Well, we'd overestimated how many genes make us up. It's now calculated that the number of human genes is somewhere between 20,000 to 25,000, far fewer than first assumed. To put that in perspective, it's actually fewer than Daphnia, the water flea, which has about 31,000. It's far fewer than even a carrot, which has approximately 32,000. So the number of genes that an organism has little to do with the complexity of the organism. And the Human Genome Project has allowed to investigate our genome in great detail and understand how all the genes interact to make the proteins inside human cells. But why do all this? Why spend all that money and all that work to get a list of nucleotides stretching 3 billion bases long? Well, it's going to help us predict and prevent diseases. By looking at the human genome or somebody's genome, it may be identify the likelihood of somebody developing various diseases, whether it be cancers, heart disease, or the likelihood of developing mental illness. And based upon that likelihood, or that prediction of you developing an illness, you could then receive advice on how you can adapt or change your lifestyle to prevent such diseases. But having a genetic likelihood of disease does not mean that you will suffer it. A lifestyle change could reduce the chances of you getting that disease dramatically. It can also mean that you might receive treatment to prevent the disease, and medicines particularly designed to stop those diseases from ever beginning. But that's not all good news. How would you feel knowing that you may well develop a disease by your mid-40s? The stress levels that could cause, the panic, and the way you may actually live your life instead. Would you be employable? Could you get life insurance? Could you get a mortgage for your house? If other people can get hold of your genetic information and realise that you may get a disease early on in your life, you may be discriminated and be prevented from certain jobs or insurance. And then there's the idea of genism. The idea that you are what your genes dictate you to be. So nothing that you will do in life will be valued because you will be labelled according to your genes. Nevertheless, the information is there. So what we're going to do with it, or what scientists hope to do with it, is that it's going to allow quicker diagnosis, to find faults in people's genomes. And that may allow them to develop better treatments, which may involve gene therapy, to ultimately cure those diseases. And the curing of such diseases will prevent discrimination or genism or stress over your genetic makeup. It will allow people or scientists to determine how effective drugs are. Not all drugs treat everybody in the same way. Some people seem to have resistance to certain drugs, which is why some drugs can cure some people of cancer but not other people's. And it's all down to genetics. So identifying which people are going to respond well to drugs is going to save time and money and save lives. It will also allow scientists to develop new drugs targeted against faulty genes, which may reduce side effects and again be faster, more effective and save money. So the potential of the human genome is massive, certainly in terms of medical science.